Hi, Arjan. Sorry for the inconveniences. Nice to meet you. My name is Medina. I'm assistant professor in the Magallanes University in Punta Arenas, Chile. And today, think about of exploring machine learning algorithms and protein language models to solve or to make classification system for enzymes or ECCB. It's a very similar topic with the previously talked. So, for a context, um, the enzymes plays a very important role in the life in general in biotechnology or in different situations. So, for example, in different biological process and decreasing the energy to make the injections, so when to make the reactions. In general, is a very important topic for protein engineering approaches. Cake is one of the most relevant repositories from the information of the enzymes, and the use of enzyme classification system, or call it enzyme numbers, is very important strategies to classify the information of the system or the function of characteristic. There are different numbers, different levels, or different information about them. So in other case, machine learning algorithms is very well strategies to understand how to develop different predictive models, how to understand the pattern recognition strategies, or in other case, reinforcement learning in case of game theory, for example. But in particular, in the use of biological or computational problem tasks associated, for example, with protein engineering problems, the machine learning could be applied in classifications or predictive model regression, for example, in classification for functional classification system, gene annotations, biological activity in the case of peptide activity, for example, or for example, more specifically, with single peptides. And in the case of regression tasks, for example, the prediction of different properties or characteristics like activity, stability, or expression. So in the case of pattern recognition, also could be applied, for example, for pattern associated with family functional characteristic or to guide the design of limitations. And combine predictive model with reinforcement learning, it's possible to improve the performance, adding information of wet lab experimental or new information obtained from the labs. So the problem is when we obtain protein sequence or when we want to apply machine learning algorithms in protein sequence is how it's possible to represent numerically the information in the protein, because the machine learning don't work with sequence. We work with text. We work only with numerical strategies. And there are different strategies based on feature engineering, for example, and more, in this case, it's possible to apply, for example, classical encoding strategies. For example, more classic, one hot encoding is very well common, and codification, amino acid encoding based on physicochemical properties, the use of transformation strategies, for example, based on digital signal processing, Laplace transformation, among others, to obtain representations in the signal space or the frequency space representation. And actually, there are a lot of posters with lateral language processing or large language model that is very well used to represent the information and create numerical representation strategy based on pre-trained models in large language models. So, as alternative, it's possible to use the information of the structural sequence or the structural protein to represent image, for example, as coordinates in the atom in the space. And also it's possible to use or to build graph structures based on the protein structure. For example, nodes represent atoms or represent residues, and the edges represent interactions, energies, or distance between atoms or between residues. So as a particular case, it's possible to use graph structure to represent the protein-protein interactions network or protein-ligand interaction networks. But, okay, how is possible? Because we obtain functional classification model, different model to solve different tasks or different protein engineering applications, but how is possible to combine the independent models with in a particular data-driven approach or in a particular strategy to improve a particular property or characteristic? So, for example, Actually, there are two very well-known strategies to solve the improve or to improve the protein, the characteristic of the proteins. Rational design is and directed evolution. Directed evolution is based on the natural selection process, and rational design is based on the knowledge of the information, the protein, knowledge of the structure, knowledge of the characteristic. But the combination of two methods is possible to use semi-rational approach based on a double-sided mutagenesis duration. And in the case of combine this method, this information with computational biological statistic method, for example, it's possible to create combined approach. But the use of machine learning algorithms is possible to create data-driven approach to add the information of new information. So, but the problem is, okay, I make mutations, I make change in the protein sequence, but the protein is still the enzyme and maintain the function of the enzyme, for example. In this case, we try to evaluate, incorporate our classification system in our data-driven platform to improve the specific properties. So the question is how to solve the classification system. In this case, the first step is I have an unknown sequence. This unknown sequence is as an enzyme or is not an enzyme. It's an enzyme. Okay, I have different easy classification numbers. And in this case, for example, you can see the first level. Then, in a hierarchical strategy, obtain the classification for the first level. Then, the second level, make classification, the third level, and enable. So, in this case, different predictive model or classification model for each level. So, in our approach, is asset only uses the sequence information. In this case, combining different pre-trained models available to create numerical representation strategies. You can see in this picture that it's possible to obtain the protein sequence and then apply different numerical representation strategies. For example, pre-trained model like Vepler, ISMB, ProTrans, or different others, the use of also physical-chemical property to characterize or to encode the amino acid sequence, and the use of fast Fourier transformation to create single space representation. Then obtain the latent space in the cases of the pre-trained model and create different individual datasets with the protein sequence encode or the enzymes encode. Then we propose a generalizable pipeline to train predictive models, or in the case of for the protein task, protein genetic task, and you obtain different datasets. You explore different machine learning algorithms, classical machine learning, with different combinations of behavior parameters, and also explore different CNN and long short-term memory architectures in the case of deep learning. So once we obtain the different models, we explore what is the better model using different classic metrics like precision, in this case, recall, F-score, and with a statistical approach, we select what is the best strategies. Then, with a different combination of machine learning algorithms and numerical representation strategies, we apply genetic algorithms to optimize the tuning process, but to create a optimized individual model. And different individual model create an unique assembled learning model or assembly process. This is, for example, different people talk in different language and different opinion for different papers to join in a unique response. So we explore, in the case of enzyme classification system, that with different tasks, with different performance, and in the case of protein language model, achieve better performance in comparison with amino acid encoding strategies or fast Fourier transformation strategies. So then, making assume in the natural language processing methods, Vepler or ECCB plus ARN methods achieve better performance in comparison with the other methods. You can see in this picture that there are differences in comparison with this method, and also we explore training and testing performance to evaluate the overfitting in this case. Then, okay, we have that pre-trained model is a nice strategy, but what is the strategy to train predictive models? We explore different architectures based on CNN and long short-term memory, and different classical machine learning algorithms. And in this picture, you can see the overfitting rate. The overfitting rate is a metric that is possible to obtain the multiplication between one loss per training dot testing. And in here, you can see the majority of the CNN architectures achieve better performance, but tend to overfitting. So in the case of KNN at random forest, also has better performance, but in comparison with other methods. So in here, we obtain then our selected approach. We choose 10 individual optimized model. And you can see in the training strategies and the representation strategies, then architectures based on deep learning in CNN architecture and representation with pre-trained models, the majority. And you can see the performance in training and testing process and the overfitting rate. Then the combination in a unique assembled model improved the individual performance, decrease the overfitting between training and testing process. And some discussions. Actually, we create this method based on a second approach, combine it, is comparable with different state-of-the-art methods. I'm sorry, I don't compare with the previous method, sorry. But we need to compare it. And also, we have comparable with a classical structure-based method with similar performance. And the method is actually we obtain a PGA model for first, second, and third levels, and we are working in the fourth level. So the performance in here, in average, because there are a lot of models, 98% of precision for the fifth level, 92% of precision for the second level, and 87% precision of the third level. And in the case of fourth level, we have a problem because there are small data sets, and it's not possible to generalize the behaviors of for the classification system. And we're exploring two strategies to work with low-end data sets, data augmentation methods, and semi-supervised learning to increase the generalizable of the classification models. So, but the question is how to incorporate this classification system in a data-driven platform. For this, we have a target the enzyme as angiotensin enzymes, and we explore how to improve the solubility of these enzymes, and we explore the similar strategy to develop a predictive model. In this case, we can obtain this model. We achieve a performance of 0.82 of Pearson coefficient, and we explore the landscape reconstruction for these enzymes, and we explore what sites in the mutations improve the solubility. Okay, first section, mutations improve the solubility. Second question, what mutations don't reduce the stability or maintain the stability of the protein? Increase, filter the sequence. Then the third problem is, okay, there are mutations that from the phylogenetic point of view is not possible to classify or are deleterious. And using evolutionary coupling, we develop a epistatic model to obtain the zones or the mutation that increase the performance, not decrease the stability, and maintain a benefice mutation. And finish with the classification system, we filter the mutations that maintain the function of the enzymes. And with this, we have a full data-driven platform with our methods. And our knowledge, the different funding institutions, CEPIP, ANIT, CADI, Universidad Magallanes, CIDG, and the part of our groups, professors PA, and Gabriel that's in here. And for your attention, thank you so much.
Thank you. Thank you. I guess
the most classical approach would be just to go into homology and ask topic annotation and compare the machine learning methods against traditional way of doing it.
Is appear similar results, but
Where you annotate a new sequence to the closest hit.
yeah.
The number down the database say that you're not fooling yourself.
Oh, sorry, I don't understand.
No.
No.
Okay, let's talk later.
Wait.